So good afternoon to you all.
Bonjour.
My name is Anne-Sophie Pezzino. I am a lecturer in psychology. I'm also a psychologist and neuropsychologist, and I work on rare genetic disorders. I've been doing that for the last 10 years. My name is Hugo Monori. I'm a speech therapist at the hospital in Rouen. I'm also studying for a master's in psychology. We are going to present where research stands on rare genetic disorders, and we're going to talk about the Mipwanel syndrome and the cognitive and behavioral impact of it. Now, for at least 15 years now, rare genetic disorders have drawn more attention. Why? Because we are trying to better characterize clinical profiles, and also because they are models that are very good for exploring neurodevelopmental mechanisms, specifically in the case of neurodevelopmental disorder. Why? Because our genetic origin is very well characterized often, not always. It's not the case for all disorders, but these disorders allow us to reduce the heterogeneity, the etiological heterogeneity that we often come across with idiopathic cases of these disorders, disorders for which we don't know the reasons. So, in 2018, we have a study by Mitchell, which underlines the use of these models to link the cause between the genes, the impact on the brain, cognition, and behavior. And this approach was a bottom-up approach. It means that we follow the whole pathway from the genetic disorder right up until the behavioral disorder, going through the cerebral stages, cognitive stages, and behavioral stages. This approach means that we can identify developmental trajectories, and they are as diverse as there are the number of humans on Earth. Make no mistake. And there is an excellent researcher, Carmela Smith, who underlined the importance of working on developmental trajectories, atypical trajectories, to better understand inter-individual variability between individuals and intra-individual within one person.
As you know, there are many phenotypes in those disorders. The neurocognitive approach is very interesting in that we center on functions such as attention, language, memory, or executive functions, and we try to identify all underlying mechanisms. Those mechanisms may be vulnerabilities, and they will help in the child's development. In other words, this perspective is both developmental and dynamic, and we try to understand the complex interactions between brain maturation, environment, and learning for children, but also for those when they reach adulthood. So, development should be understood as a dynamic, open system that keeps changing, and a system that's also very sensitive to early interactions and inter-individual variations. So we need to think about the neurodevelopmental disorders in what we call a neurointegrative model. So we take into account the development trajectories, but also genetic influences of each of our models. So neurocognitive approach. So what is a neurocognitive development? Well, those are all the changes in the brain throughout a trajectory. So from prenatal period to adulthood. So it appears that there are constant interactions between genetic programming and early environmental influences, if you will. So genes are going to guide the major stages of brain maturation, but their expression in turn is going to be modulated by the environment. And this is what we call epigenetic mechanisms. So a rich environment is going to promote brain plasticity. This was mentioned by one of my colleagues. A rich environment is also going to optimize neuronal connections and therefore is going to stimulate cognitive functions. Brain development also follows a progression which is not linear and is not necessarily superimposed with biological stages. So first you have neurogenesis, formation of neurons, then neuromigration in the frontal cortex, then the synaptogenesis, when all neurons are going to create links or linkages, and some neurons are going to create a sheet of myelin, which is going to help and increase the speed at which information is transferred so much that we are going to be, as an individual, we're going to be able to adapt quicker to new situations. And these stages are going to create the basis of complex cognitive functions, especially what we call executive functions. So our ability to plan an action, to inhibit a behavior, the ability to be flexible or not flexible in a situation, and also social functions. Those two last ones are really the result of maturation of the frontal network and frontoparietal network. They continue and develop till adolescence. So what I'm saying here is that even when you're older, you are still able to adapt, and that's certainly a glimpse of hope. Now, neurocognitive development is, as I said, a dynamic process which depends on brain maturation, but also by the experience experienced by a person. Now, those neurocognitive or neurodevelopmental disorders are often the result of abnormalities in the maturation of the central nervous system. The reasons for that etiology is multiple and diverse. This is nothing but the reflection of genetic, epigenetic, environmental factors. And in the case of TNDs, these disruptions can happen rather early in life, and we talk about the cascade of cognitive, emotional, and behavioral consequences. One of the main challenges when we study those DNDs, one of the major challenges, as I said, is that they are so diverse, very heterogeneous. We talked about intersyndromic variability. In other words, there are symptoms that are going to overlap between different diagnoses. For example, attention deficits, executive function deficits you're going to find in ADHD, but also in ASD. And you may find intersyndromic variability that will be intrasyndromic, between different patients. In ASD, for example, some individuals will have severe individual disability, while others will have preserved or even above average cognitive functioning. When those functions are altered, they will of course have an impact on behaviors. It will translate in less cognitive flexibility. So behaviors is going to be more rigid. You know, people say, oh, you are very stubborn and you have quite a temper, but it may so happen that those patients don't do that on purpose, if you will. Also, if you have language acquisition deficit, then it may translate in difficulties in interaction, social interactions. This is going to be changed by what we call cross-cutting factors. For example, sleep disorder can aggravate attentional difficulties and emotional instability. In the same line of thought, high anxiety can interfere with working memory or decision making. You may also have avoidance behaviors or opposition behaviors. Then if you have sensory disorders, which is very common in TND, it may lead to sensory overload and also intense behavioral disorders, agitation, etc. So many interactions here. And I'm not done. I told you it was complex. Also, we have medical comorbidities. Epilepsy is one of them. We may also have psychosocial conditions, as we call them, psychosocial factors. Parental stress is one of them. And you can also have, on top of everything, psychological disorders, for example, anxiety or depression. So all this taken into account, all those comorbidities are going to either exacerbate certain difficulties or mask an underlying disorder. And that's why, and let me insist on that, we really need to have a dynamic, integrated approach when we work on rare TNDs. And when you're a clinician, you should always have that type of approach. That MIT1L syndrome, I think, is a very good and concrete illustration of those connections between genetics, brain development, and cognitive functioning. So MIT1L is essentially a transcription factor, is an essential transcription factor, and obviously, in case one of those processes are disrupted, is going to have an impact on language, executive function, emotional regulation, and social skills. Thing is, all those clinical manifestations are going to vary considerably. So they're going to be very specific to the person with that syndrome. So it adds, if you will, an extra layer of complexity. Now, for you to better understand the impact of abnormalities, I had a look at well-characterized genetic syndromes. For example, Williams syndrome. So it's associated to an atypical neurocognitive development. Language is preserved, but there are visuospatial deficits, hypersociability, difficulties in regulating emotions. Same goes for Down syndrome. Moderate to severe intellectual disability. A lot of variability in language. Some of those patients are going to talk and others will not be able to do that. Then we also have you also have another type of syndrome where again you're going to have different level of language acquisition, but then you're going also to have vasospatial disorders, and episodic memory, event memory is also to be impacted. Now, I will talk about the syndrome of Dravet, maybe a different name in English. Those children have very severe epilepsy, and again, moderate to severe intellectual development disorders, and we have a hard time understanding their language deficiencies because we need to adapt to their seizures, and they also have motor disorders, spatial disorders, and often there's also as a comorbidity a autistic syndrome. So I'm not giving this list to be comprehensive or to bore you to death, but each syndrome has a particular cognitive signature, if you will. In some cases, capacities are well preserved, but in others it is not the case. Now, those profiles change with time. I love developmental theories, and so I think that a cognitive profile is not set in stone. Therefore, it is key to conduct longitudinal studies, to study them over time. And their skills, they can change and they can improve throughout their life. We just need the right keys to understand and help them develop such skills. You see that there are also many variabilities within the same syndrome. And again, this interest is to look at developmental trajectories and not, again, think that the profile is set forever. So working on rare syndromes is interesting in that we create models and then we better understand what's really at play. And this is what we talk about a transdiagnosis approach. It's very dynamic. We don't only center on genetics, on cognition. We try to work together. And I think we see more and more research groups doing that, especially in that very room. It's very cross-cutting. Now, if we read literature with Hugo, who did his PhD work on MID1L. On MID1L, we have 40 to 50%, well, that's what we read in literature, but half of those individuals have intellectual deficit. It's very heterogeneous. Some have no diagnosis, no deficit. There are other comorbidities according to literature. ADHD between 40 and 50%. A disorder on autism spectrum, about 40% of those, they are on the spectrum. Also, in literature, a number of areas are not even studied, especially on cognition and memory. So there are very few and far between studies on this. And those profiles, of course, are associated to specific behaviors. So aggressivity, heteroaggressive behaviors. And again, I would like to really underline that it's when those individuals are stressed. They are not born aggressive. Such a thing does not exist. It's only under stress. Stereotypical flapping, rocking, something that you find on the spectrum, 43% according to literature, hyperactivity associated to an attention disorder. And there are also a few studies, but it's not very clear. Apparently, little tolerance to frustration, but there may be also a link with executive functions. Then, when it comes to what we call the modulating factors, so I'm not going to pick up again on eater disorders with hyperphagia, increased risk of obesity, oral disorders, food selectivity, difficulty in chewing. We find anxiety in about 20% of those young individuals, and when I talk about young individuals, again, maybe adults as well. We find also sleep disorders, neurosensory disorders as well, not very published in literature. We find auditory and tactile hypersensitivity. It's quite marked. And this is based on literature, but by the way, it's also something that is reported by parents or carers. So, in other words, MIT1L, I think, is a very good illustration of how complex neurodevelopmental disorders can be when they are of genetic origin. So, we really need a detailed, multidimensional assessment. And let me state the obvious, we do not have such a detailed, multidimensional assessment tool to date. Hence the interest on the MIT1L project, which was created by Dr. Cosimo, and I think she deserves many thanks for that, and thank you for integrating us in your team. Now, I give the floor to Hugo. Well, my colleague already said quite a lot, but obviously we need more work to characterize cognitive and behavioral profiles. The Stéphane Rondeau already mentioned that not two ADHD are the same. So when you say that you have ADHD, it doesn't really help you understand what's at stake. And for attention deficit, there are different types, different components, and so you really need to go in depth and check whether our patients have difficulties on all items or not. And then based on that assessment, we can propose different adaptations and different interventions. Very few studies also studied modulatory factors, and there's not necessarily a correlation between those modulatory factors and cognitive functions. Therefore, we designed a study whereby we tried to really understand that in the context of neurodevelopmental disorders, try to see if there are cognitive specificities associated with MIT1L syndrome. Wanted to know as well if it would change over time and according to the different adaptation abilities that you have. So that's our program. We really centered on cognition, behavior, and also we screened for sleep disorders. So there were 10 tasks that the participants had to go through, really depending on their age, their fatigability, etc. So we wanted to explore the different cognitive functions, so attention, visual attention, so when you really treat one information amongst other visual information, auditive and divided attention. Divided attention or split attention is when we treat many information at the same time. That's what you do at the same time. You're listening to me, you're reading the slides, so you're doing different tasks. Now, we had different tasks for different items. There are also different tasks for executive functions. Executive function is, if you will, a toolbox whereby we adapt to new complex situations. It really groups together a number of functions and capacity. It has to do with memory, our ability to change our point of view, focus on some information and forget others to reach an objective. So those executive functions are key and help people to adapt on the daily. We talked already about language and communication, but we didn't really test that because speech therapists are really the experts in that. So that's why there were not too many tests in that respect. We also tried to test reasoning ability and also processing speed. Those tests, by nature, have many limitations. It's not necessarily a good reflection of real life. So it's important to say that from the get-go. But we know that also it's very sensitive, where it's very fatigue-dependent. It's also very dependent on the context, because usually you take those tests in the hospital, so it's a source of anxiety, etc. So you need to take those results with a pinch of salt. And that's why we also designed many questionnaires. We gave out tons of questionnaires to parents, and I know it was a lot of work for them. But in order to have a more comprehensive picture, if you will, better understand what happens to those children, but also to their parents on the daily. So questions on attention, executive functions, attention. We also wanted to assess the modulatory function factors, modulatory factors being anxiety, sensory integration, and sleep. Now, those are a few tests. There are about 10 of them, so we're not going to go through all of them. So first we had those matrices. So you have a sequence, and in those 5 items, you need to find the one that's logical. The symbols help us measure the processing speed, and so what we measure is how quickly the child is able to find the symbol. So we assess processing speed, and we also assess a number of visual and spatial behaviors. Then you have the visual puzzle. So you have this form, this shape, and the child should point at the 3 elements that make up the puzzle. And there are some pieces of a puzzle that are well rotated, if you will, well positioned, and others are not. And so it's really for us to assess whether the child is able to abstract and reposition that shape mentally, if you will. Then there were a number of tasks. We asked the subject to group shapes and group animals or garments, etc. We also had a test on divided attention. Subjects were asked to identify an image or a picture and a sequence of sounds, and so they had to find the right target. So they had different things to deal with at the same time: sound and pictures. And auditory attention was tested with colors and sounds. And then there were distractors in that we were also giving cues that had nothing to do with the test. Also, to assess opposite roles, we instructing them to say to when they would read one, and vice versa. So they needed to design different strategies, and also it's a way to test motor inhibition, because when you see one, you have to say two. So there we go. And then you had go, no go. It was a way for us to see how they have cognitive inhibition, how flexible they were, and whether they were able to control themselves and do what was required from them. And this figure here is rather abstract, and so we ask subjects to replicate that figure. We can assess many things, and so we were able to see what were their planning strategy, whether they would start with the outline, whether they would start with the from the outside in out, they would start with details, or would start with a big picture, etc. So it has to do with praxis as well. When we start with the outline and then you go towards the details, usually it gives a better result. So many tests, as I said, and I suppose that for families as well, they had many questionnaires, so they had a lot of them on their plate. So on attention, hyperactivity, behavioral disorders, but also on learning. There was also a questionnaire on executive functions, tackling different executive functions of the daily life. There was a questionnaire on communication and language, so we sort of stole the thunder of speech therapists here. Then anxiety, sensory integration, in order to demonstrate hypersensitivity or hypersensitivity, and we know this may change over time and throughout the day. And there was also a rather simple screening test for sleep disorders. With that, we had tons of data, preliminary data. So we need time. Analysis is in progress. So for the time being, we have not much that we can report, but a very high intra and intersyndromic heterogeneity. 22 participants, and not two were the same, obviously. All of them were very different in the way they interacted with the test and how they went through those tests. Most of them were had a high fatigability. They were vulnerable to anxiety. They were very sensitive to noise, and you know that in hospital it's rather a noisy environment. So this was something that made those tests more difficult for them. There were also some sensory features, especially some subjects were very sensitive to smells. And definitely there were some attentional and executive frailties or vulnerabilities, if you will. So what we wanted to mention as well before we wrap up is that families were fantastic. They were very much involved. I know that it was very hard. Some of you had to drive many hours to come to the hospital. I know it's difficult for you to let your child go through so many tests, and then you have to fill in all those questionnaires, and and we know that it comes on top of so many other things. So you're very involved, and really you deserve a heartfelt thank you. Now, before I conclude, when we study rare genetic syndromes, we have a valuable opportunity to fine-tune or refine our understanding of typical development trajectories. We better understand connections between genes, brain development, cognition, and behavior. Behavioral symptoms are always the result of something. They're not here for no reason, if you will. So it really helps with understanding. Obviously, it's important to continue and study this mid-1L syndrome because it's still poorly documented, and so this warrants more studies. We need tools that are standardized in order to be able to compare studies, to also compare studies designed in other countries, and so that also the data produced are more statistically reliable. We also need more longitudinal studies with long-term follow-up, so as to better describe their cognitive, emotional, adaptive profiles over time, and see what are interventions with effects they have on cognition but also behavior. And I think that's it. I think that's me. Thank you very much for your attention.